So I'm excited today to present part of the project that I'm doing in my PhD. The main reason I'm actually excited to present today is that November is actually Pancreatic Cancer Awareness Month, and having this result, as in trying to find T-cell targets and doing antigen discovery on pancreatic cancer, I'm presenting it in November. I'm hoping that it would provide more momentum towards this goal. This was a collaboration between our lab, Purcell Laboratory in Monash, and SciEx in Canada. So pancreatic cancer has multiple subtypes. The main subtype is pancreatic ductal adenocarcinoma, which has as a majority of the pancreatic cancer cases are pancreatic ductal adenocarcinoma. It has the 8th rank in terms of incidence, 3rd rank in terms of cancer-related death, and only 11% of the patients would survive after 5 years of receiving the diagnosis, which is one of the poorest survival rates among all types of cancer. If the patient is diagnosed earlier in the stages of the disease, they can benefit from conventional chemoradiotherapy options and surgical options. But majority of the patients are actually diagnosed after local advancement or distant metastases to other tissues and organs, which makes our conventional approaches a bit less effective towards this disease. So the hope is to actually find an immunotherapy option for this disease. Precisely the approach that we are pursuing is a T-cell based immunotherapy approach. And well, T-cells, primarily those based on a T-cell receptor or a TCR, recognize their targets as a short peptide on the cell surface, presented to the immune system in a specific way that I would describe in detail later. But the end goal of immunopeptidome studies is to have a rational design for the T-cell based immunotherapy. So as I mentioned, pancreatic cancer has one of the poorest outcomes and poorest prognoses among all types of cancers. That has to do with a multitude of factors, including having a very suppressive and cold microenvironment, which makes T-cells inept or less effective when they see a pancreatic cancer cell in the tissue. So our goal is to find an effective T-cell target, a peptide, that we can actually teach the T-cells with that when they see that peptide or epitope or target, they can actually effectively eliminate the cancer cells and essentially eliminate the tumor or control the tumor at least. Or in other words, the end goal is to change the picture of T-cell meeting a cancer cell in pancreatic cancer from something like this to the T-cell standing up to the cancer cell and effectively eliminating them. But what is an immunopeptide? What are immunopeptides? Immunopeptides are teeny tiny peptides derived from source antigens through specific machinery in the cell and are presented on the cell surface to assist immune cells, precisely in this case T-cells, with immune surveillance. These peptides are presented in the context of a protein complex with a family of proteins called major histocompatibility complexes, MHC, which has two classes, class 1 and class 2, with their own unique characteristics like length distribution, the type of motif that they accept in terms of arrangement of amino acids. To study immunopeptidome, we first need to immunoprecipitate the MHC peptide complex, isolate the peptides and clean them up, run them on the mass spectrometer after getting the peptide list after research, characterizing the peptides, and if they actually follow the specific characteristics that we want or we expect them to have, like the length distribution, like the motif, as I mentioned above. If they follow these characteristics, then we follow our pursuit with target discovery. And these targets have different flavors, you may say, as in you might actually go for tumor-associated antigens, mutated peptides, PTM peptides, or, for instance, endogenous retrovirus-derived peptides. But regardless of what flavor these peptides have, you can actually have a different approach towards immunopeptidome as well. As in saying, there are potentially immunogenic peptides that are upregulated on the tumor cell, but are not upregulated or are not present on the healthy tissue or even healthy distant tissue, as in healthy pancreatic tissue or healthy skin, for instance, in a pancreatic cancer patient. So if you can find an immunogenic peptide that is upregulated on the tumor cells, regardless of its flavor, then you have an effective immunotherapy approach, or you're closer, at least one step towards it. You can have different quantitative assays to look for or to quantify the immunopeptides. You can use a targeted MRM or PRM approach, for instance. You can even do it with a DDA acquisition approach, data-dependent acquisition approach, or a data-independent acquisition approach. Each of them have their own pros and cons. If you go with the targeted approach, you're limited by the number of targets you can study. You need to have a prior knowledge of the targets as well, but it is a very sensitive assay. If you go with the DDA approach, you're sort of leaving it to the stochasticity of the DDA method, to pick it up and fragment the peptide to acquire a spectra to identify it. And it has the issue with the missingness and missing values in your output metrics. In a DIA approach, you have a bit of headache with the complexity of the analysis in terms of you have chimeric spectra, spectra that is just full of fragments. It makes it a bit complex, but it has the added value of being more sensitive than DDA, at least, and having a more complete matrix, quant matrix in the end. So that's why we chose the DIA or data-independent approach. If I want to give you an analogy about what DIA is, imagine you went for camping. You saw this beautiful scenery. You wanted to capture every detail of this beautiful trip. Precisely this scene, but you have a camera with you that can do a long shot, 180 degrees long shot, but it is going to be a pixelated and low definition photo. Or you can choose a very close angle of the scenery and take a high definition photo of it. So you try and see that the photo that you took is very pixelated. It's not very well defined. But just to try your chance, you start taking overlapping short angles across the horizon, and later on when you go home, you can actually overlap them, and well, you have the scenery at hand. The photo is there, the panoramic view is there. Or you can actually do something else, as in use the secret mode that this camera has, and hold your finger on the shutter button, and while you are sweeping over the horizon, you just take a motion picture, multiple overlapping pictures that are just after each other. And that actually gives you a better idea of whether this bird was here and here, or it actually was here only and only. This is literally an analogy of what an MS1 is and what a tandem mass spectra in a DIA approach is. This is the variable or window-based, overlapping window-based swath, and this is the scanning swath that SciEx recently released. The variable window xenoswath and ZT-scan DIA. And you see that this idea of taking multiple motion picture spectra gives you a very deeper and enriched view of the analytes in your mixture, as opposed to the variable window xenoswath. So we are going to do immunopeptidomics, quantitative immunopeptidomics. We have a collaboration with SciEx as well. They mentioned to us that there is this ZT-scan DIA acquisition mode. Let's try how it works. We said, okay, show us some results. So we started this collaboration and a friendship that we later on developed with Ihor and Catherine. And the data they showed us is ZT-scan is achieving the same or equivalent quantitative performance, like in this case, the 10%, less than 10% CV or coefficient of variance protein groups are similar in ZT-scan and the xenoswath, but in a 10 times faster run, as in 3-minute versus 30-minute gradient. Then we said, let's try it in-house, as in we need to see it by ourselves. We need to see it within our data. And we started running a serial dilution of K562 digest, human proteome digest, tryptic digest, in a microflow setup with 5 microliter per minute flow rate, 1 minute, 3 minute, 5 minute, and 15-minute gradients. And in almost all of the cases, and I'm just showing you the total precursor count, in almost all of the cases, and combinations of parameters and injection amounts, ZT-scan is acquiring more or achieving a higher depth of the present analytes precursors in this case. If I want to show you this with a bit more detail, in our case, we were thinking, okay, the amount of sample that we have usually in immunopeptidome is something between 1 to 5 nanograms per injection or per sample. It's a very low or minuscule or low input type of sample. And if we actually want to do a 5-minute gradient, what is our gain in terms of precisely quantified precursors, for instance, or precursors that are identified or quantified with less than 10% CV? What is our total gain? And we see that ZT scan in this case is giving us something like 60% higher, or like 40%, if I remember correctly, or 35% higher total depth. And in terms of CV less than 10%, we see a similar pattern. And across all the parameter sets, this is what we see, that ZT scan is giving us a tighter CV or coefficient of variance, which shows us a higher precision in acquisition in quantification as opposed to variable windows. The other parameter that we needed to make sure we are getting it correct is the quant accuracy, which in simple definition is how close your measured values are to the actual value that you wanted to measure, and or in a more sophisticated way, what is the distance or how close the median of the measured values are or is to the actual value. And there's a brilliant design, the LFQ bench type experiments, that you mix up multiple triptych digests of multiple species, in this case 3 species, human, yeast, and E. coli, in different ratios, and you try to measure or visualize or assess the accuracy. You make a reference sample with equal amount of human, yeast, and E. coli, and then the next 6 samples are reciprocal ratios of yeast and E. coli, while keeping the amount of human constant in all of the samples. And then you already know that, for instance, comparing it, the N7, the sample type 7, with the reference sample gives you, or should give you, 1.5 times more intensity in E. coli protein, half intensity for the yeast proteins, and same intensity level for human proteins. So your ratio is already known, or expected ratio is already known. And then you measure this ratio for each protein separately and plot it, and you see whether the median of these measured values for the more abundant species, the human reference or the human sample, or human proteome, and the lower abundant species, how close is it? How close the median of those measurements is to the expected ratio? In almost all of the parameter sets that we tested, ZT scan is providing a higher accuracy compared to variable window xenoswath. And as this is shown here, as in your median is closer to the expected ratio. xenoswath is doing a really good job, but ZT scan is doing it better, is what I would describe in this part of the results. The other value of doing an LFQ bench study or experiment is that you can actually, while you are searching these 7 experiments together, you can search a K562 as in a complete human sample that you know it hasn't been touched by any E. coli or yeast peptides or proteins, and search it alongside with the other 7 with the same settings, as in FASTA data as in library-free search, or a human yeast E. coli library-based, spectral library-based. What you expect is that proteins that are identified and quantified in this sample are only from human, but in reality, you will get human and a bit of yeast and E. coli, as in few yeast and E. coli proteins are also identified and quantified in this sample, which gives you an idea of empirical calculation or measurement of your false discovery rate. they shouldn't be there, so they are falsely identified and quantified. So we did so for all the parameters that we tested, as in all the methods that we had. And in almost all of the cases, we see higher depth of identification and quantification achieved by CT scan, while the FDR at precursor and protein level is lower. Just to show you one of the examples in a closer view, this is a 250 nanogram LFQ bench injection with also a 200 nanogram K562 searched with them. Methods had 7 millisecond dwell time in a 5-minute gradient separation. As you see, the ZT scan method is actually giving us a deeper view of the precursor and proteins identified, while having a lower FDR. And when we turn on the match between run in our Diane settings, this actually stays the same, as in we identify more precursors and proteins as opposed to variable window swath, while having a lower, ever so slightly, but lower false discovery rate. Now, I like microflow coupled to mass spectrometry, but I'm more of a big fan of nanoflow setups. As in, the type of samples that we have is very low abundant types, or a limited input type of samples. We have only a few nanograms of peptides in each sample that we can run. And nanoflow separation gives us a better separation method for our acquisitions or for our assays. In this case, we used a 15 centimeter Aurora Elite Anoptics column in a 15-minute gradient separation at a flow rate of 115 nanoliter per minute on our M class, coupled to the Xenotoph. We injected 5 nanogram or 15 nanogram of K562 digest. And first, we see something close to 10 to 20,000 more precursors using the nanoflow setting, and 1,000 to a bit higher than 2,000 protein groups with nanoflow compared to microflow. We also see the increase or that gain that we achieve with ZT scan as opposed to variable window xenoswath. And precisely, for instance, with this small sample, like 5 nanogram injection, you see that 20% more precursors identified and quantified using the ZT scan acquisition. Now, everything is ready for going through the immunopeptidomics work. So we grew our trustee PANC1 cell line, which is a very well-known pancreatic cancer cell line used in many studies in vitro to study pancreatic cancer. We grew them in normal condition without adding any specific cytokine or anything else, just normal media. We pulled down the MHC class 1, cleaned the peptides, ran them on the on our Xenotoph, and searched it with both variable window xenoswath and ZT scan DIA. Searched the data with Diane 1.9.0, and it was in a library-based search. The spectral library is is a DDA high pH fractionated library that I made previously in maybe 2 years ago. After characterization and realizing that the peptides that we identified with ZT scan actually are following the normal characteristics of these peptides. We searched for specific tumor-associated antigens, and here I'm leaving the endogenous retrovirus-derived peptides out of this presentation. So the setup is the same thing as I mentioned above. ROI lead, 15-minute gradient, 150 nanoliter per minute. The amount that we injected is equivalent of 2.5 million cells LWET, or peptides derived from 2.5 million PANC1 cells, or 25 million PANC1 cells on column. First, we see that from this very low-yielding cancer type, we are seeing 12,000 peptides identified in a single run, which is just amazing. I don't know how to describe how I felt when I saw this this depth of identification in this assay. In terms of the length distribution, we are not seeing any sort of biases that is induced by the ZT scan acquisition. It is still following the expected length distribution of class 1 peptides. But the actual added value of ZT scan in this case is you're having a tighter CVs with ZT scan at equivalent loads and other parameters. As in, with 2.5 million cells, you're getting tighter CVs with ZT scan. With 25 million cells, you're getting, again, tighter CVs compared to variable window xenosweth. In terms of other steps of peptide characterization, we see that previously actually showed that xenosweth identifies the same peptides, or it doesn't affect the peptide characteristics in terms of binding characteristics or motif characteristics compared to DDA acquisition. And here we are showing that ZT scan is working in terms of peptide characteristics similar to variable window xenosweth. And we are identifying, or we are seeing, all the expected motifs of the present allotypes in PANC1, which is A2, A11, B38, and C1203. Now, targets. I'm giving you two examples of the interesting peptides and as in tumor-associated antigens that we saw. And the first one is this peptide from this very famous tumor-associated antigen PRAME, which you're seeing the ZT scan spectra, this beautiful, clean ZT scan spectra on the top versus the library transition on the bottom, on the negative axis. And, well, seeing how Diane is performing well in isolating or extracting the signals across a very narrow peak and showing the coelution of the fragments in this run for this peptide. The other example has to do with another tumor-associated kinase, in this case, TTK. And this peptide, as you see the same beauty in terms of the mirror plot, how clean the spectra, the ZT scan spectra is compared to the library transition list. But the added value of ZT scan in this case, you see that the interfering fragments are now resolved with ZT scan acquisition, which is actually provided by that very narrow 1m/z isolation windows that ZT scan has. Now, to summarize, we actually showed the higher speed, the improved speed, precision, and accuracy of proteomics assays using ZT scan DIA on our Xenotov 7600 Plus. Also, we showed the ability of doing immunopeptidomics with ZT Scandia on the 7600 Plus. The need that we have for a sensitive assay to study cancer epitopes is actually very well met. And in terms of pancreatic cancers, as it is, it has very low number of mutations, as in low mutational burdens. There are very few neoepitopes that are identified in pancreatic cancer, but we still could identify cancer testis antigens, ERVs, and phosphopeptides from this type of cancer. We recently submitted a manuscript showing, as I mentioned above, comparing Xenoswath acquisition, variable window Xenoswath acquisition, compared to DDA in minuscule samples, like 1 million cells per pellet, and injecting 1/6 of that. And thank you. I would actually encourage you to go to LinkedIn and watch this beautiful clip on Pancreatic Cancer UK, which shows how we don't need just hope. We need the action towards finding a solution for pancreatic cancer. And it's very clear that this work couldn't have been done without a close collaboration between multiple teams, SciEx in Canada, SciEx here in Australia, and the team we have in the lab, precisely my supervisors, Tony, Shri, and Pat.